AIDS has been discovered almost 4 decades ago, and since then, it has claimed over 35 million lives worldwide. Have you ever wondered why it is a disease without a cure? Why is it so difficult to rid a patient of the infection in spite of treatment options available to them? Today, combinatorial therapy will let you live a longer life even after being infected with HIV. But even then, one cannot live a life with a complete eradication of the virus. Current drugs only slow down the progression of the disease, but there still thrives a sleeping fraction of virus that remain undetected by the drugs. This sleeping fraction, in the absence of drugs, will resurface and cause infection. Therefore, we need to find ways to circumvent this shortcoming and kill the sleeping fraction so that HIV infection will reach a complete cure in the near future. Nowadays, different therapies target to cripple the virus itself. But in contrast to that, I do not target the virus, but its direct environment. My approach to the problem is not by looking into the virus, but around it. Like humans, viruses also need certain molecules called proteins in order to function. These proteins make sure that activities within the cells are carried out without obstruction. In my research, I look into the environment of the virus and see what its protein requirements are. And like I mentioned earlier, by targeting these proteins, I try and cripple the virus and stop them from functioning. More specifically, I target the protein anchors that the virus uses to take over and cause damage to the human DNA. This can be compared to something as crocheting. I need this hook if I want to add a different yarn to my existing pattern. Similarly, HIV uses protein anchors as hooks to enter the human DNA. These protein anchors additionally carry signatures, and these signatures are needed by the virus for the proper identification and insertion into human DNA. In my research, I developed compounds or drugs that render these signatures unrecognizable to the virus, and thereby presenting alternative options to patients. With my approach, I hope to develop efficient therapeutic strategies which are better than the existing ones and thereby help for a complete eradication of the disease.